This is the Forensic Biology Screening Workshop presented by the National Forensic Science Technology Center. The first part of this workshop deals with laboratory orientation. The laboratory orientation lecture will be split into 5 segments: safety, clean technique, reagent preparation, use of basic equipment, and quality assurance. We will now go over the National Forensic Science Technology Center's Environmental Health and Safety Program. The NFSTC's Environmental Health and Safety Operations Manual is divided into 5 separate sections or plans, and they are listed below as follows: the General Office Safety Plan, the Laboratory Safety Plan, the Emergency Action Plan, the Chemical Hygiene Plan, and the Biohazard Exposure Control Plan. The list below discusses both general and operational rules that apply to the chemistry and biology laboratory sections. No eating, drinking, applying cosmetics, or adjusting eye contacts in the laboratory areas. No running, jumping, or horseplay shall be permitted. All spills will be cleaned up immediately. Always wash your hands before leaving the laboratory. Leave the laboratory clean and organized at all times. No sandals or open-toed shoes shall be worn in the laboratories. Know what you're working with and know where the necessary spill kits can be found if a spill does occur. Personal clothing worn in the laboratory should offer protection from splashes and spills from chemical or biological fluids, and should be easily removable in case of an accident. Lab coats are not to be worn outside of the laboratory areas. Inhalation exposure is one of the 4 modes of entry for chemical exposure. Sniff testing should not be performed, and never pipette by mouth. Always use a bulb to pipette. Now let's go over an emergency action plan. Know your evacuation routes. Know the fire extinguisher, AED, drenching hose, and first aid kit locations. Know what to do in case of a fire emergency, if there is a laboratory injury or accident emergency, a bomb threat emergency, chemical spill or exposure emergency, a biological spill exposure emergency, a hurricane emergency and how to prepare for it, and other natural disaster emergencies and their preparations for them. This is an example of an emergency evacuation plan. Notice the green lines are showing how you can reach the exit quickly from every room in the laboratory, and also where the drenching hose and eye wash stations are and where the fire extinguishers are. Now let's go over the chemical hygiene plan. This is also known as the Hazard Communication Plan. Consult the MSDS before using any chemical. Be familiar with and use personal protective equipment like the PPEs, example gloves, safety glasses, and lab coats. Utilize chemical hoods to limit exposure. Use and dispose of all chemicals properly. Ensure that all containers are properly labeled, example with the NFPA labeling. Store chemicals appropriately. Be familiar with the Chemical Hygiene Plan section of the NFSDC's EH&S Program Manual, and contact the safety officer or laboratory staff for use or disposal assistance if needed. Now for the Biohazard Exposure Prevention Plan. Treat all laboratory areas as being a potential biohazard. Be familiar with the Biohazard Exposure Prevention Plan of the NFSDC EH&S Program Manual. Use personal protective equipment at all times when handling biohazardous materials. Dispose of all biohazardous waste in properly designated containers, example red bags and sharp containers. After performing daily tasks, always disinfect work areas with fresh 10% bleach solution. Wash hands after removing gloves, and use biological hoods when necessary to limit exposure. Let's go over clean technique. This refers to the laboratory practices employed to reduce the risk of contamination. One type of contamination is the transfer of DNA. This can occur from analyst to sample, from environment to sample, and between samples, which is known as cross-contamination. Contamination can adversely affect the outcome of a case. Therefore, it is essential that the laboratory have procedures in place to limit, recognize, and address contamination. We will now go over the clean technique with respect to work surfaces and equipment. First, you should clean before contact with evidence, between evidence items, and after evidence processing is complete. A common practice is to place glassine whey paper, Kim wipes, butcher paper, or bench coat paper on the bench top to act as a barrier while processing evidence. Change paper and clean bench top between items. Instruments like forceps, scissors, scalpels, and tube openers should be cleaned just prior to use. If sterile disposable instruments are used, these should be opened just prior to sample processing and discarded after one use. Use a 10% bleach solution or a commercially available sterilizing reagent such as Sidex Plus. After an item or surface is cleaned with bleach, rinse with purified water or alcohol to prevent the buildup of sodium hypochlorite crystals. Instruments or equipment cleaned with bleach should be rinsed to avoid corrosion. We will now go over the clean technique with respect to reagents and sample processing. When possible, prepare reagents in bulk. Each analyst is then provided with an aliquot for his or her individual use. Reagents should be kept closed when not in use. Samples should be processed individually. Only one sample should be opened at a time. Unknown samples should be processed separately from reference samples. Processing may be separated by time and/or space. Here are some good lab practices with respect to the clean technique. Gloves should be worn throughout sample processing. At a minimum, gloves should be changed at the completion of each step of the process. If gloves become contaminated, discard them and replace with new ones. Lab coats should be worn at all times while processing evidence. Reagent preparation is a very important part of the laboratory screening process. A laboratory should establish quality control procedures that include preparation, use of both purchased and prepared reagents, reagent container labeling, documentation, quality control testing, storage conditions, expiration dates as appropriate. For the purposes of this workshop, all reagents will be prepared by the instructors. Students may need to perform simple and/or serial dilutions as directed by the instructor. Here is some important information dealing with dilutions. First, simple dilutions. A unit volume of a liquid material is combined with an appropriate volume of a solvent or diluent liquid to achieve the desired concentration. The dilution factor is the total number of unit volumes in which the material will be dissolved. For example, a 1:5 dilution is achieved by combining 1 unit volume of diluent plus 4 unit volumes of the solvent. Serial dilutions are a series of simple dilutions used to quickly amplify the dilution factor, starting with a small initial quantity of material. The source of dilution material for each step comes from the diluted material of the previous step. The total dilution factor is calculated by multiplying the individual dilution factors of each step. Here is an example of how to perform serial dilutions. To perform a 3-step 1:100 serial dilution, first, the initial step combines 1 unit volume of the starting solution, for example 10 microliters, with 99 unit volumes of solvent, 990 microliters, and that is equal to a 1:100 dilution. The next step, the 1 unit volume of the 1:100 dilution, is combined with 99 unit volumes of the solvent, now yielding a total dilution of 1:100 by 100, which is equal to a 1 to 10,000 dilution. The third step, the total dilution, would be 1:100 by 10,000, which is equal to 1 to 1 million total dilution. Use of basic equipment. Students will be shown how to use all necessary laboratory equipment. Example, the alternate light source. You will ensure that safety glasses are worn when using this piece of equipment. Now let's go over quality assurance. Here are some of the components of a quality system. Use of adequate facilities, use of validated methods, use of properly calibrated and functioning equipment, establishment and implementation of a corrective action when needed, proper training and continuing education for personnel, use of appropriate evidence control procedures, use of established analytical procedures, reports, participation in proficiency testing programs, and participation of a laboratory accreditation program. Quality assurance, often known as QA, consists of all the planned and systematic actions necessary to demonstrate that a product or service meets specified requirements for quality. The planned and systematic actions are documented in the Laboratory Quality Manual and Standard Operating Procedures, the SOPs. For the purposes of this training, all trainees will be required to document each test performed following the procedures provided. Procedures established by your laboratory system may vary slightly from those provided in this training.